Welcome back, everyone, to Vox Markets. My name is Paul Hill, and I'm delighted today to be able to speak to Alastair Smith, CEO of Avacta Group, a leading developer of breakthrough cancer treatments, biotherapeutics, COVID tests, and diagnostics. So welcome, Alastair.
Hi, Paul.
Big congrats on this morning's comprehensive business update. There's obviously been a lot happening over the last 12 months, which kept you guys pretty busy. But before we sort of try and go and unpack those details, can you just quickly give us a bird's eye view of the company's two leading technologies, the Affimer and Precision platforms?
Yeah, of course, with pleasure. So let me start on the Precision platform. So in straightforward terms, the Precision platform allows us to take a chemotherapy and reduce or remove the side effects. So Precision prodrugs are chemotherapy without the side effects. The way in which that works is that we make a small chemical modification. So the Precision technology is a small chemical modification which renders the drug inert, inactive and harmless until through the circulation it gets into the tumour, where there is an enzyme that's specific to the tumour which clips off the precision chemistry, activates the drug in the tumour, and therefore spares the healthy tissues. So that's the Precision platform. I'm sure we'll talk more about that as we go through the interview. And then the Affimer platform, well, I'm sure all your listeners are aware of antibodies. You know, a third of all drugs in development now are monoclonal antibodies, but there are drawbacks to antibodies. You know, they're expensive, they're complex, they're relatively difficult to manipulate and put into different useful, therapeutic formats. And an Affimer is effectively an alternative to antibodies. It's small, it's robust, it's quick and easy to produce. We can produce all sorts of clinically interesting formats, so stick two together to hit two drug targets at once, for example, and so on. So you should think about the Affimer platform as an alternative to antibodies that can identify a target, lock onto it, and if it, for example, blocks the biology of that target, you have a therapeutic. Or if it can identify that target and give you a signal, you've got a diagnostic.
Yeah.
So the applications of Affimers, as we've discussed before, are very broad across therapeutics, diagnostics, and other fields.
And these are just enormous markets. I mean, the bread and butter sort of like first line cancer treatment, just, it's sort of like, it's still chemotherapy across the lot of the world because one thing that people forget about chemotherapy, it is actually effective. It's just the toxicity, isn't it, that gives it. And I saw some sort of figures that, chemotherapy is worth sort of like, $50 billion. And I saw antibodies are actually even a bigger market, I think, per year as well. So, I mean, they're just huge, these markets you're addressing.
Yeah, they are huge. I mean, just to pick up your point about chemotherapy, you're absolutely right. These are effective drugs. Many of them have been around for a long time and are generic now. They're off patent. And the problem with chemotherapy, almost all chemotherapies, is not efficacy. It's the tolerability for patients because of the side effects. So if you can remove or dramatically reduce those side effects, you can open up markets, expand markets, and really give benefit to patients.
I mean, you even have patients, I understand, who effectively respond well to chemotherapy and then have to be taken off the treatment because of the worry of the danger of actually having contamination elsewhere in the body, it seems.
Yeah, absolutely right. I mean, I'm sure we'll talk about AVA6000. And in the case of doxorubicin, it is heart damage. So there is a maximum of 6 cycles of treatment before the damage, the systemic damage to other organs, particularly the heart in the case of doxorubicin, is so severe that the patient has to be taken off the treatment, even if there is benefit in terms of tumour shrinkage. So you're absolutely right. Yeah.
And then just sort of like just big picture wise, in terms of sort of the IPR protection, patent protection. I mean, is anybody anywhere close to this? Because I understand the Affimer technology came out of, I think it was your department at Leeds University, wasn't it?
That's not quite right, and
okay.
Absolutely wrong of me to claim credit for that. No, so I mean, both of those platforms have a long academic incubation, if you like, of 12 to 15 years. So the Affimer platform originally came out of Cambridge, an academic called Paul Coferigno, who then did move to Leeds.
Apologies.
And we acquired that the Affimer platform when Paul was at Leeds, and then the Precision platform has been developed in academia at Tufts University in the US, in Boston, by a very talented chemist called Bill Bachovchin. So both of these platforms would be very difficult to replicate. The Precision platform, other people have tried to use enzymes in the tumor to switch on therapies to avoid that systemic toxicity. But the particular enzyme we're using, FAP, I believe I'm right in saying that nobody has successfully harnessed that. And it's because unless you get your mechanism absolutely specific to that enzyme and not any closely related enzymes that are all around the body, then you end up with your drug getting switched on all around the body again. So what Bill has managed to do is develop some chemistry that's absolutely specific to the tumor. So the drug only gets switched on and activated in the tumor. And then in the case of the Affimer platform, there are other non-antibody therapeutic platforms out there. Very few that are, that are really effective. But within the therapeutic space, it's really about clinically differentiating what you do. Okay, so it's about using your platform to develop drugs that are clinically differentiated, that give patient benefit and therefore commercial value.
Yeah, I mean, I think what's important again for investors as well is to realize these two platforms have numerous application areas, and you say in diagnostics and therapeutics. And if we just sort of now step through, I mean, this is just one. Obviously, the first one, which is in most investors' lips, is the rapid lateral flow antibody test, isn't it? Can you just take us through that one particular use?
Yeah, absolutely. So the rapid antigen test is a form of immunoassay. So people should think about a pregnancy-style test. So where you see, one red line or two red lines or blue lines, depending on whether it's positive or negative. And what happens inside those tests is that there's an Affimer, which when it sees the antigen from the virus flowing past, captures that antigen, and that's what creates the line. So the Affimer is sitting in a line, waiting effectively for the antigen to pass, captures it, and that's how you get the red line on the test.
Does it attach itself to the spike protein?
It does in our case, yes. So there are two antigens that you could use with this particular coronavirus. One's called the nucleocapsid antigen, one's called the spike antigen. There are reasons for choosing one or the other. Our view, if the listeners are interested, is that the spike protein, because it's on the outside of the virus, is a stable, soluble, fairly robust protein. And of course, you have to remember what you're trying to do is detect the spike protein that's originated in the respiratory tract and has got out of the respiratory tract into the nose, onto a swab, into the test. If that antigen is not robust, if it falls apart and sticks to things, never gets out of the nose or never gets onto the test, then you're going to have very poor sensitivity. So we chose to work on the spike antigen because of its robustness. And I think that's been borne out. I'm sure we'll talk about the clinical data in a moment, but that's been borne out in the excellent clinical data that we've seen very recently for the test.
So effectively, the reagent, what it does is it attaches itself to the spike antigen and then tells everybody that actually we've got an alien body here, and that's the reason why it's positive and negative, and the accuracy is so good. And I'm guessing going forward, can you just take us through the different variants? We've obviously got the South African one and the Brazilian variant, but I'm guessing just sort of top level wise, because it's engineered. And it's a human sort of protein, your Affimer, then you can actually just tweak it as a vaccine can be tweaked as the protein spike moves and changes, can't you?
Yeah, so we have demonstrated that variants that have appeared to date, the Affimers that we've used in the test already identified those variants. The very new ones, the South African, the Brazilian, we're still doing the work to demonstrate that. We expect that the same Affimer will detect those as well. But yeah, your point is absolutely correct. It only takes us a few weeks to generate an Affimer. So if we want to know in the future, if there was a variant that was very different and you couldn't detect it with the existing tests that are available, we could very quickly, in a few weeks, generate a new Affimer against a new variant. Or indeed, let's think beyond COVID, infectious diseases in general, generating Affimers that identify and detect other infectious diseases, future pandemics, and so on. There's a very clear platform here for rapid response. And the amount of work, the real bulk of the work that we've put in over the last 6 months has been around making these tests work with complex nasal samples, which, as you can imagine, are fairly complex, and I won't describe them. I'm sure everyone can figure it out. So that, those are difficult samples because of the nature of them. But now that we have a test that works well with those types of samples, you can drop a new Affimer in really quickly. You don't have to redevelop that test. So yeah, it's an interesting point.
It's future-proofed, effectively, isn't it?
Exactly. Yeah.
My wife, she works at a school, and there's about 1,200 kids there, etc. They're all going back on the 8th of March. And Boris has said for his, he wants everybody tested twice a week, and there's about 10 million. If you add up all the kids and the other teachers, about 10 million. So that's 20 million tests they're going to have to do, etc. Now, the problem that they've had, obviously, they're using lateral flow tests currently, and the accuracy is a bit, you get 50% of infections from people who aren't showing any symptoms. So how do you see this one solving a lot of that issue? Because obviously the accuracy of the test is first rate, I understand.
Yeah, so I mean, we could talk about this for hours. Really, there's an awful lot involved in everything you've just said. But so first of all, just to touch on the performance of the test, this is critical, and we talked about it when we announced those results. So once someone becomes infected with the coronavirus or any virus, there is a period of time through which the virus begins to replicate, and what's called the viral load, the amount of virus in the body increases. Your immune system begins to deal with that, and ultimately that viral load will fall away. Now, in the middle of that process, the individual is infectious, whether they have symptoms or not. At the point where they have a high viral load, they could be shedding that virus while speaking, laughing, and so on, and by touch on surfaces, but more with coronavirus through sort of the respiratory tract. So what is absolutely essential for lateral flow tests is that they can identify those infectious people, whether they are symptomatic or not. And without going into lots of technical detail, we know from PCR measurements what that level of infectiousness is. And the clinical data that we announced last week was for that bracket of individuals, patients who are clearly infectious. Even if their viral load is at the bottom of that range, they're still infectious. And yes, you're right, the clinical data that we presented was excellent, 97% sensitivity within that range of infectious individuals. Now, the mistake I think that some people have made is trying to compare lateral flow tests to PCR across the entire range of infection. Yeah. Because once that virus, the viral load drops, you will not detect that with a lateral flow test. It's not as sensitive as PCR, but that's not what it's intended for. So it's only intended to identify infectious people. And the whole concept of rolling it out to schools, to workplaces, universities, and so on, relies on the individuals being able to run the test, so that the sampling has to be straightforward. And the nasopharyngeal, the brain poker, as people call it, that's very difficult for an individual who's not well trained. Even when you're well trained, it's very difficult on children. I did it on my own kids. It's very difficult to push that swab that deep into their nose and throat. Whereas our test is the anterior portion of the nose, so the nostrils just a sort of centimetre or two in, so not unpleasant at all. So that's a big plus in terms of
absolutely.
Children, older people, and so on. So yeah, excellent performance, relatively easy sample collection. Those are the key things. And the final point I would make is that it's not just the sensitivity that's important here. Yeah, of course, you need to pick up as many of those infectious people as possible so they can isolate and you reduce the spread of the disease. But the specificity is really important, because if you turn up a lot of false positives, if you have a low specificity, you get false positives. You're then isolating people who are not infectious, may not have the disease at all, and then you've got an economic, a social and economic cost through unnecessarily isolating people. So specificity is really important as well.
Yeah, I mean, just to underline that, I mean, when they get a positive at my wife's school, the whole year group then has to quarantine. So that's 100 kids. Just on timelines, when we should be able to buy these tests, do you think, Alastair?
No, it's the obvious question. So we are now in the process of, so we've got a body of data, which is the first clinical data that we've obtained, which is obviously very encouraging. And now we are going into what's called a full clinical validation. So that's the clinical data that you need in order to be able to CE mark the test. There's other work that you have to do alongside that around stability and so forth, reproducibility and manufacturing and those types of things. And then all of that forms the package that you use to CE mark the test. And we should, if all goes well, have that package of data by the end of this quarter and be able to CE mark for professional use by the end of this quarter. There are a lot of things to get right. I mean, being realistic, but we are very close now. Yeah.
Okay, good. And then I'm guessing if the schools want 20 million of these tests just on the road each week, how much volume can you give them?
Yeah, no, that's been something we've been working on for many, many months. And, there's a lot of companies in the UK that have collaborated in order to get that capacity in the UK up from where it was pre-COVID. So this is not just Avacta by any means. There's a whole list of companies that are involved in increasing the manufacturing capacity. So as we launch that product, we will be able to access a few million per month, and that'll ramp up as we deliver the demand that arrives. So we can scale that in the UK. We're looking abroad as well, in Europe and elsewhere, to add to that capacity. Within the UK alone, it is growing now, the manufacturing capacity. So it's difficult to put a precise figure on, but I'm sure there will be 10 million a month quite comfortably, and higher than that, certainly, as the investment that's gone in is implemented in new equipment, new manufacturing processes.
Yeah. And I'll just highlight to investors, I mean, even if you guys only did 2 million a month, that's sort of like, I don't know, I'm just putting a figure out, £5 each, that's 10 million a turnover. And none of that's in the broker notes at all at the moment. So that should be all upside. Just finally then, just on the crystal ball, once we're through the pandemic, mass vaccinations, etc. I mean, I saw a note come out from Boston Consulting Group the other day, and they are still reckoning we're going to need hundreds of millions of tests each day. And presumably the authorities are going to migrate to the better tests and the more reliable tests, aren't they?
Yeah, no, absolutely. So my view is this, that vaccination rollout is going extremely well in the UK, less well in other places. I think generally speaking, the peak demand for antigen testing will be this year and next year. So I think that's when the peak demand is. Beyond that, I read that same report. I agree that there will be testing requirements through to 2025 and beyond. And the reason I say that is that this disease is not going to go away. It will ultimately be like testing for influenza. So there will be a long-term market for testing. And I agree with your point that only the best tests will capture that long-term market. And that's part of the reason that we've spent time, getting a high-performing test, because long-term, when the market is smaller and supply isn't the issue, it will be the better tests that really capture that long-term market.
Yeah. So just turning now to the second application area of one of the platforms, the provision platform, with the Pro-doxorubicin, with the phase 1 trials. What are the sort of key milestones that investors should think about for this year?
Yeah. Okay. So I mean, I think this is, as exciting, if not more exciting, and long term, there will be requirements for cancer treatments for many, many, many years. That's clear. So what we've done with the precision platform is we've taken initially a drug called doxorubicin, which we mentioned earlier on, a very effective chemotherapy, but with serious cardiotox, serious heart side effects. We've modified that with the precision chemistry. And what we've seen in animals is that we get a dramatic increase in the activation in the tumour compared to any healthy tissues, the heart in particular. So the safety in animals is dramatically improved. And without going through all the data, the survival in animals, reflects that. So we filed what's called a CTA, which is the clinical trial authorization in the UK before Christmas. And we very recently got approval from the MHRA to go into that phase 1 trial. So the phase 1 trial is in 2 parts. The first part is what's called a dose escalation study, where you identify the correct level of dosing. And then the second part is a dose expansion, where you look at particular cancer types, and that will probably be 2, maybe 3 different cancer types. So we should be dosing the first patient in the dose escalation phase in the middle of this year. And the key time point will be before the end of the year. Actually, we should see the first what's called PK or pharmacokinetic data, and that'll tell us whether the precision platform is activating the drug in the tumor predominantly compared to healthy tissues in human. Yes. We know it does in the animal models, but does that also do that in human? And that is enormously important, because not only does it give us a valuable asset that we can develop with the Pro-doxorubicin AVA6000, but it means that precision chemistry is working in human, and we can translate that then into a whole pipeline of other chemotherapies, which we've got waiting. We've already developed a version of the drug Velcade, and also oxaliplatin and paclitaxel. So we have the next 3 in the pipeline, ready to move forward rapidly when we see those data.
Yeah, and just on scale, again, I'll just point to investors. I mean, more people die, I think, from tumours and from cancer than they do from COVID in the whole, in the world. And I think what you're saying is that, chemotherapy, which is a sort of $50 billion business already, per annum, this technology, the precision platform, will be able to increase the usage and the applications and the patients who can benefit from chemotherapy, which is pretty effective in terms of sort of like its performance wise, and therefore the markets actually should increase in theory as well.
Yeah, I think that's a really, really good point. So when investors think about the precision platform, this is not just about trying to grab part of the existing doxorubicin or Velcade or oxaliplatin market. If you can reduce the side effects and make a safer version, then you can start to expand the market by treating patients for longer, so more than 6 cycles. So if we could treat patients for 12 or 15 cycles, there'd be enormous clinical benefit. And of course, you've tripled your market size straight away for that drug. But also you can start to think about treating patients who have some form of comorbidity, a weaker heart. I mean, patients are not treated with doxorubicin if they're older than 65, I think is the figure. And yet you could open up that market. Most cancer patients are over 65. So, it's not just about trying to grab part of a market. It's about creating a much larger market as well.
And then just as if everything goes to plan, at the end of this year, we get the green light, it works, it shows it reduces the toxicity in the heart, particularly from doxorubicin, etc. Then presumably, I mean, doxorubicin has been around for 40 years. It's a well-known drug. So, phase 2, phase 3, I mean, it's obviously all got to be safe, but the medical scientists and the authorities know how this drug performs. And if it's shown that when you combine it in this pro-drug format, that actually it makes it safer, you should see accelerated approval for this to bring to market, because there's literally millions of people around the world who can benefit from it.
That's right, yeah. So our understanding from the regulators at this point is that because, exactly as you described, this is a well understood drug that's been around a very long time. In fact, the phase 2 study would be what's called pivotal. That would be the study that would get market approval. You wouldn't need to go on to a phase 3 as you would do with a new drug entity. So that also accelerates the time to market, which is commercially very interesting as well.
Yeah. So just moving now on to the third application, it reminds me a bit of Mozart and his symphonies. This does. You sort of like compose the next one. But when we talk about sort of the Affimer therapeutics with the your lead drug, the AVA 004, and other ones in development, how do you see those going? I mean, again, we're just talking enormous markets. I mean, immunotherapy and stuff like that is multi-10 billion.
Absolutely. I mean, these are world-class technologies. I mean, I'm not embarrassed to say that. We have world-class technologies here, and the potential for them is enormous.
Yeah.
So we've talked about the diagnostic applications with Affimers, and there are, of course, as you say, significant therapeutic, and we're focused on cancer immunotherapies.
Yeah.
Working with partners in other areas, but our particular focus is around cancer immunotherapies. And when we spoke the other day, the point I made to you is that you don't want to simply copy what other people are doing, but do it with an Affimer. You want to do something that Affimers allow you to do that's very difficult with other platforms, that gives you clinical differentiation, gives you something that is therefore adding benefit for patients and therefore is commercially valuable. And the two areas that we are focusing on are what are called bispecifics.
Yes.
So the fact that we can take two Affimers and put them together, one hitting one drug target and another hitting a different drug target, so that you can treat two pathways at the same time.
Yes.
That's not trivial to do. We can do that relatively straightforwardly with the Affimer platform. So the bispecifics is an area that we're focusing on. I probably won't go into the science of what those targets are now. We'll get bogged down, but that's one area. And then the other area is what we call a TMAC. So this is this is a fantastic concept that we came up with Bill Bachovchin actually at Tufts. And it's a form of what's called a drug conjugate. So you take an Affimer and you chemically attach that to a chemotherapy.
Yes.
Now, normal drug conjugates would just use the Affimer to target the tumour. There would just be a biomarker, a cancer biomarker, and it would target the tumour and deliver that chemo warhead.
Yeah.
What we've been able to do, because we put the precision chemistry in the linker, is we've been able to take something like an immunotherapy, so a PD-L1 inhibitor, and attach that to a chemo warhead. And then that gets released in the tumour because the precision linker is cut by the enzyme that we discussed. So you release a warhead, that starts to kill the cancer cells with a big inflammatory response, which attracts the immune system. And you've got an Affimer immunotherapy there to support the immune system when it gets there. So I realise the biology is complicated, but that is completely novel. The concept of having an immunotherapy as part of a drug conjugate is completely novel, and we've patented that concept. And you get the synergy then between the two different therapies, which should give a longer lasting benefit to patients.
Yeah, I mean, I think investors really sort of take some, interest in that particular concept, the TMAP, because, bringing the two platforms together into one format where you get the benefits of the chemotherapy at the same time as you get the benefits of being able to enable somebody's immune system to be able to attack the tumour at the same time. I mean, not only could you get shrinkage, you could actually get almost, unheard of, but you could almost get a cure for a lot of really late stage and advanced cancers that frankly, people aren't diagnosed and have got very little chance of survival in the long term.
So let me just touch on that point very briefly. And it is a bit more biology, but it's really interesting. So this is why large pharma are interested in talking to us about the TMAC platform. If you look at an immunotherapy on its own, so just a single immunotherapy like a PD-L1 or PD-1 inhibitor, only about 20% of patients respond to those.
Yes.
80% of patients don't respond to those single immunotherapies. And the reason is that the immune system is not already inside the tumour.
Yes.
Okay. If the immune system's inside the tumour, then blocking PD-1 or PD-L1 will support the immune system and you get a response. That's the 20%. What we're doing is we're using the warhead to inflame the tumour, to attract the immune system in, and synergize that with, so you expand the patient population that should respond to that, because you're giving the the stimulatory effect to get the immune system in, and using the checkpoint inhibitor. So there's a lot of detail, obviously, in the biology, but yeah, it's a fantastic concept. It's obviously, if we're honest with ourselves, more challenging to develop. It's longer term, but that's the sort of thing that's really clinically differentiated, that adds value for the company and for shareholders, and is likely to be able to do big deals in the future.
Yeah, I mean, again, just as a sort of checkpoint for investors, I mean, the world's leading immunotherapy drug, Keytruda, is set to sell about, I think it's $20 billion a year at the moment. So that's just one of them. I mean, again, it just puts the scale of this, the applicability to it, and just coming into sort of the next symphony for Avacta. I mean, you're in-house, well, the diagnostics and also your licensing. I know we'll probably talk about that at a later date, but just give us a wet investor's appetite for the openings there, because I mean, again, you've signed some unbelievably good deals in terms of, sort of like LG Chem and all this kind of stuff. It'll take a lot of time to go into, but just sort of top level, just give us a quick view of how that's developing.
Yeah, certainly. I mean, I think that's an important validation for external observers, investors, and generally, because what you can see is pharmaceutical and large biotech doing deals to work with the Affimer platform and the precision platform. That gives you some confidence that that the science is solid and there's real potential for these technologies. Out with what we're saying, of course. So yeah, we have deals with Moderna, with LG Life Sciences, Daewoong, Point Biopharma, ADC Therapeutics, and maybe I'll just touch on the Daewoong Pharmaceuticals one. It's another really interesting one. Daewoong, I think, is the largest by revenue South Korean pharmaceutical company. I believe that's true. It's a big company. And what we are doing with them is combining the Affimer platform with their stem cell platform.
Yes.
To generate stem cells, which obviously have a therapeutic benefit themselves, to generate stem cells that will actually make the Affimer inside the stem cell and spit them out into the body as they are doing. So the Affimer supports the stem cell therapy by modulating various inflammatory pathways. Now, that is a completely novel next generation stem cell therapy, which we wouldn't want to carry out off our own balance sheet. So these types of partnerships really give us an opportunity to to push the barriers and do things, frankly, on somebody else's balance sheet that can further the platform. So that one, I think, is a particularly exciting one. But yeah, we have a number of very meaningful, significant upfront payments and future milestone payments.
These are in all, different therapeutic areas. It's infectious diseases. It's in autoimmune conditions. I mean, stem cell. I mean, these are just massive markets on their own, and hugely growth as well.
Well, that's why we have to remain focused. If we tried to spread ourselves across all those markets, I mean, you can imagine it would be a mistake. But working with partners, we can support them to use the Affimer platform and the precision platform in those different markets, whilst we remain, tightly focused on the programs that we've talked about. Yeah.
And if you get to sort of like the Mozart equivalent of sort of 41 symphonies, have you got enough resource and bandwidth to be able to sort of like continue composing?
Well, it's a sensible question. Obviously we are well funded. We have a very strong balance sheet. We've used that to expand since last summer, which has been absolutely critical for the points you've just made. There is a lot going on, and you need the bandwidth to do that properly. So, we've put in place a head of product development, a product development manager, product managers, operation directors, and the diagnostic business. And in the therapeutic business, we've appointed a Chief Development Officer, Neil Bell, who's absolutely outstanding. And with him, a head of CMC, head of translational science, clinical operations, and quite a large, what I would call virtual drug development organization, outsourced service providers. So we are doing a lot with these platforms, but we have been able to resource that properly so we can deliver on schedule.
Brilliant. And again, I just really highlight to investors. I mean, I'm a bit embarrassed because I'm a professional investor and I haven't really looked at Avacta, you know, really until the last sort of 6 months, and I've missed the sort of the applicability and the scale and the magnitude of the two leading, you know, the platforms associated with. But even so, your market cap is currently 450 million, and frankly, if any one, and this is my view, any one of these leading sort of technologies commercialized, you know, fully, you know, these are billion plus, you know, opportunities without any problem. And I just again point to people, have a look at sort of what's happened to Novavax this year, just on vaccines. It's got a market cap of 20 billion. Have a look at sort of like Exact Sciences, again on 23 billion. Nobody ever heard of BioNTech, and that's on $27 billion. So, I mean, who knows what the, you know, what the scale of it is, but all I can say is that I really hope you're going to be successful, because you know, there'll be, you know, huge gains to come through it, and there'll be, you know, huge therapies. So just finally, before we sort of wrap up, what in terms of framing the next 12 months, then we've talked about sort of like you know, sort of the Pro-doxorubicin. We've also talked about you know, sort of the COVID testing, etc. Is there anything else of sort of, I mean, you know, you've got so much going on. I presume there's going to be some more sort of news flow coming through.
Yeah, absolutely. Yeah, I mean, to summarize really, COVID rapid antigen test, CE mark as soon as possible. Obviously then sales and revenue on the back of that's this year and onwards. The AVA 6000, that we should have the first patient dosed with that middle of the year, as we discussed, and data before the end of the year, then the dose expansion next year. So all very significant value inflection points, you know, for the company. Then we will also deliver a range of clinical candidates to take forward into the clinic in 22 and 23. You know, that's important. If we don't deliver those clinical candidates to the development team, they can't be taken into the clinic. But then also a range of commercial deals on both sides of the company, you know, commercializing both the COVID-19 testing as well as the non-COVID tests that we have, and licensing deals and partnerships on the therapeutic side. So there's a huge amount of technical as well as clinical as well as commercial milestones and news flow over the coming 12, 18 months.
Brilliant. Okay, well, thanks very much for your time, Alastair. I mean, you know, you get a tick in the box from me, that's for sure. I mean, I think personally, I think it's fabulous technology with a huge addressable market, and I really do hope, say, the Pro-doxorubicin, you know, comes through, and then, you know, my wife will be cheering like to the rafters if you can deliver the lateral flow testing in sort of like, you know, a couple of months' time. So thanks very much for your time, Alastair.
Yeah, thank you very much.
Thank you.